Hello everybody and welcome to this iFormulate webinar on formulation of microbials for agrochemical applications, and I'm joined today, and my name is Jim Bullock from iFormulate, and I'm joined by my colleague David Calvert, who you can see on your screens there. We'll be running for about 45 minutes plus question time today, and we're very much looking forward to talking about this subject. So welcome. We're going to be looking at microbials for agricultural uses. What are they actually used for, and how are these products used? We're going to be talking a little bit about the benefits on of using these microbials and the market growth that the market has seen for these products, both now, previously, and in the future. We're going to be having a discussion, David and myself, on potential formulation challenges, and then David is going to be taking you through some examples of formulation of microbials, and we'll follow that at the end with a short question and answer session. Today we're keeping you muted, both your video and your audio muted, but you're very much welcome to use the chat function for any questions you may have, as well as any technical problems that we will try and sort out for you as the webinar proceeds. This webinar is being recorded, and we will be sending you a recording and a copy of all slides after today's event. I think we can start very briefly by talking a little bit about iFormulate itself. We're a consultancy which helps companies with formulation challenges, and we specialize in the formulation area. And of course, as ever, we'd be very much pleased to hear about any challenges you have in formulation. We've been going for about 13 years ago, founded by David and myself. We're combining our own quite diverse experience and knowledge with a wide range of contacts that we have in the industry. And where necessary, we will bring in associates to help us on assignments if they are needed. In the second column on this slide, you can see a list of the industry and product areas that we have experience in. I won't read that whole list out, but you can see that it covers not only agrochemicals and agriculture, but the whole range of industries that use formulation science and technology. Our own experience has been not only in consultancy, but also innovation and R&D, very much so, also in business development, in strategy, in regulations, in training and events. And on the right-hand side, you can see our contact details, and of course, you're very much welcome to contact us at any time. What do we actually do? Well, I would say what we do for our clients falls into about three main areas. Probably the majority of work that we do is consultancy, and this is normally carried out under a non-disclosure agreement, whereby a company will come to us, or an organization will come to us and ask us to carry out a particular consultancy assignment, a challenge or a technical problem, and which may be short-term, it may be long-term, in the formulation area. And we'll work to given timelines and deliverables to deliver that assignment for the client. We've also done work for industry bodies, industry-facing organizations, to help them develop strategies for formulation and their approaches to the formulation market. And in the third box, which is really what we're covering today, skills and training. So we work together with other organizations or on our own to deliver training, both public open training, but also customized training, and this training can be online, it can be in person, to individuals and to companies. So all of those areas, and again, if you have any particular training needs, we'd be very pleased to hear from you. But let's move on to today's topic, and today's topic is microbials for agrochemical, or rather agricultural applications. And I think first it's worth defining what these microbials are. And I find this particularly useful because I'm not a microbiologist, I'm not a biologist, I'm a chemist. And many formulators are. They've come to formulation from the chemistry angle, some come from other disciplines, of course. But I think it's worth defining the microbials. So the ones that we see in the industry are mostly bacteria and fungi. Occasionally viruses are used. The bacteria, and there's a drawing of a typical bacterial cell on the right-hand side of this image, but the bacteria used are typically gram-positive bacteria. That means they react to the gram staining test because they have a thick peptidoglycan cell wall layer and a thin lipid bilayer in the cell wall. So very much the majority of bacteria used as these products are gram-positive. Occasionally you will see gram-negative bacteria, and these have a thinner peptidoglycan layer, but they do have an outer membrane layer for nutrient transport. And in fact, the gram-negative products are less favoured, probably because they're a little bit more sensitive to environmental conditions, whereas gram-positive types tend to be a little bit more robust. Often bacteria used, and we'll show at least one example later, are spore forming, and these are essentially protective casings that some bacteria can form, and these can protect bacterial cells from their environment, and that can be very useful to the formulator. And typically these, just to get your dimensions in your mind really here, typically these cells are several microns in diameter. They're not usually spherical. They can adopt a whole range of different morphologies. So that's bacteria. Moving on to the fungi that are used in the industry. These are typically unicellular, but they have a particularly complex life cycle very often, and they may be formulated in the form of these spore-like conidia, tend to be a bit more spherical, or these elongated hyphae forms. So you might see fungi in a number of different morphologies in products on the market. And what are these microbials actually used for in agriculture? Well, a major category is that of the microbial bioinsecticides, and these are bacteria or fungi used to control unwanted insects or other invertebrates. The products might also include metabolites of microbes, so not only living microbes, but also metabolites. There are also biofungicides, and these are bacterial fungi typically used to control unwanted fungal infection on crops. And a third category, and an interesting growing category, is that of biostimulants, and these are microorganisms used to help promote the growth of crops and to improve yields, for instance, via supporting the colony of the root microbiome and improving the use efficiency of fertilizer nutrients. One area that I think has interesting potential for development and could be a major commercial interest if it were to develop would that be that of a bioherbicide. These are pretty rare. I believe there is at least one product on the market based on metabolites of bacteria which can act as herbicide. On the right hand side, I'll just give you one example of the way that a fungus, in this case Beauveria bassiana, can infect an invertebrate. You can see colonizing the surface, but also more importantly, that bacteria is colonizing the internal parts of the invertebrate and essentially killing it off after a period of time. So it can be very, very effective if you understand their life cycle. And here are some examples. David will probably expand, I think, later on some more of these products and organisms. But I thought these three kind of give you a range of different properties, really, in terms of the active ingredients. So the first example on that list is Chromobacterium and the product name. And there are numbers of products on the market, and there's no particular connection between ourselves and these companies. We just picked some examples from the open market just to show you what's there. So ProFarm's Grandivo is based on Chromobacterium, a particular strain of Chromobacterium. And then, in fact, although it contains cells and spent fermentation media, the product isn't alive, it's dead. So there's no living microbes, there's dead cells and metabolites, fermentation media in the product, and these are effectively the active ingredient. This one's formulated as a water dispersible granule. Moving on to Dipel, a very well-known insecticide, miticide product based on Bacillus thuringiensis, a particular strain of that. This contains, as its active ingredient, not only living cells and spores, which are alive, but also fermentation solids and the protein toxin, the actual active ingredient toxin as well, the insecticidal toxin, which David will cover a little bit more later. So it's a mixture of living cells which can generate the toxin as well as existing active ingredient in there as well. This again can be formulated as a solid, as a water dispersible granule. And the third example on this is the example of that fungus that I mentioned before, Bavaria bassianus, is another insecticide. This one has to be alive because it only works if it can infect and go inside the organism that it is trying to control. So it's very important that this product consists of living cells. And in this case, this is formulated as a water-free oil dispersion of the Bovaria cornidea. And that product naturalis comes from BioGuard. So products on the market, but why are they there? What are the actual benefits of these products? And I think it's often done to compare these products with traditional crop protection products or fertilizer products on the market, typical chemical products, if you like, that can be compared with these. And today is not really the time to go through the debate of whether any of these things are fact or perception. I think there's probably a mix of fact and perception in all of the things on these bullet points on this slide. But certainly the benefits in the market are those of safer or less toxic products, of more sustainable products which have reduced environmental impact, that potentially have fewer issues with resistance to pests, that have no chemical residues, that can mean the product can be, or the crop can be harvested immediately after treatment. They're naturally based solutions, which is an attractive thing to have in the market. And alongside that, they are very often compatible with organic farming methods. And in some parts of the world, compared with synthetic chemical products, microbials can have faster registration approvals. And it's important to realize that this is quite a rapidly growing market. And the figures I've taken from third parties here of publicly available numbers. And just as a caveat, these numbers include not only the microbial biocontrol market, but the non-microbial biocontrol market. We're not talking about that today, but that would consist of things like natural extracts. So the microbials and the non-microbials are natural products. They're all naturally based products. And these are lumped together in these market numbers. But despite that, you can see there's a very significant growth rate, 11% compound growth rate foreseen over the period 2021 to 2029, up to a really large 15 billion figure, according to Dunham Trimmer. And that's an increasing share as well as the overall crop protection market. And likewise, biostimulants growing at a similar sort of compound growth rate to about half that size in terms of its market. And again, that contains microbial as well as non-microbial biostimulants. So there's a strong growing market and some perceived and actual benefits in the market as well. So at this point, I'd like to bring in my colleague David. He'll be joining us for a short chat before he will present a few slides before the Q&A session at the end. So I think it's interesting, David. Many of us, and I think we're both in this category, come to formulating microbials with a chemistry background. So typically we formulated chemicals and used chemicals in formulation. And then we as chemists are confronted with these strange things based on biology, so microbial species. And that can be quite challenging, I think, as a newcomer to that area. What do you think are the initial challenges that we as formulators should be aware of?
Well, I've made myself unpopular sometimes with people who produce microbials by saying that actually I don't think it's any different to a traditional chemical that you're trying to formulate in agrochemicals or in any other market. as a formulator, what you need to do is to understand your active ingredient and what the sensitivities are to the active ingredient. So if you're formulating a microbial, you need to start to understand what is it, what are the active ingredients, and what are the sensitivities of that, be it a spore, be it a microbe, be it the metabolites, to the various conditions that you might be putting it through as you formulate it and as it's applied, because your job as a formulator is also to preserve it during the application, and in this case in the field. So I might be a bit blasé, but I don't think they're too much different than a normal chemical. They're just different, I guess, as everything is.
Yeah, I think I would agree in general with that. I think the issues of stability and compatibility, so you have to make sure that your active species is compatible with the co-formulants that you're using, and you would have to do that in any case as a formulator. You'll be checking out chemical and physical stability. You may have the additional challenge of microbial stability to encounter as well. I think one thing that is slightly different is that of what I call viability, and I think it's when you're formulating a chemical for application, then the chemical that you start with can only degrade. You can't increase its concentration as you go through the formulation and application process. So you start with a number and you keep that as stable as possible and as high as possible in terms of its concentration. Microbials can of course grow, and that's
Yeah.
both a good and a bad thing, because microbials can grow after they've been applied to the crop, and you want them to do that. You want them to proliferate and become active on the crop. So they're increasing in concentration. So I guess as a formulator, you've got to support that. But you don't want them growing in the can, because if they grow in the can, they're pretty soon going to run out of nutrient and die even before you come to apply them. So I think there's a slight difference in terms of viability. But I do agree with you on understanding the organism, and those three examples that I gave before. We had one example which only consisted of material from some dead organisms, if you like, metabolites and cellular matter, one of which was a mixture of living organisms and cellular byproducts, and the third one which was only living organisms. And that's absolutely where I agree with you. As a formulator, you need to understand that active ingredient very well.
Yeah, and I think, if you look at what's been going on over the last few years, the issue of viability or incompatibility of various co-formulants is really starting to be addressed by the co-formulant suppliers. And I think that's taking the formulation element forward a great deal, because they're able to say, well, you can formulate with this, or no, don't formulate with this bacteria, because it does hinder the viability, and the inhibition tests are fairly commonplace now. And I think that is in a way no different. It's just there's more information available now than there was 5 years ago. Whereas if you're traditionally formulating a chemical, that's been around for a while and people have had that information at their fingertips almost.
Yeah, certainly the knowledge base is improving. So as a formulator then coming to this, you've stopped worrying, you've understood your organism, and you're starting to plan your formulation approach. How would you do that in the case of microbials? How should you be doing that planning?
Well, again, people who've listened to me, it starts to be like a bit of a broken record, but everything starts in microbials just as it does in another formulation, with understanding what it is you're promising to deliver and having a design process for that. So we call it design for formulation, and it's applicable across all markets, and it starts out with your customer promise. So why are you developing this microbial, and what is it going to do in the final product for your customer, which is the grower? So what are you promising to deliver? And if you start with that, that then leads on to, well, what are the critical quality attributes of the formulation? And then what are the critical raw materials? And there's no doubt that the critical raw material will be the microbial, or it may be actually the metabolite, and we'll talk about the metabolites. But I think, again, the design process and starting this off is not really that different to a normal one. It's just it hasn't been done as often.
So you're saying if we paid attention to you in the past, we should be well set up to formulate microbials.
if you'd paid attention to me in the past, you might have made a load of mistakes initially, but let's learn from those mistakes. Yeah.
Well, yeah, and I think in terms of changing and developing and improving, I think one of the things that we've probably both observed in, more than 10 years of looking at these sorts of formulations is that microbial formulations have had to change, develop, or improve in those years. What sort of changes have you been most impressed by?
Again, I think it'll come back to moving on from a simple formulation um, into understanding what formulation can really do. And the challenges, certainly if you'd have talked 5 to 10 years ago for microbials, were a lot about efficacy. And they're okay in this niche, but we really, to get a bigger market penetration, they need to improve their efficacy. And I think that comes, as it does in all other formulations, it comes from how it's put together, be that structure, be that through the co-formulants, and that improves the efficacy rather than having the microbe or the metabolites doing all the heavy lifting. And you see that in other formulation areas where you add something and you get a synergistic effect, for example. And I think that will that's certainly started to move on and has impressed me a great deal.
Yeah, I think I'd agree with that. I think when these sort of products were fairly new on the market, people were prepared to put up with
Yeah.
their unusual behavior and having to take great care of them, and were accepting of challenges and trickiness in application and sort of variable quality standards. But as these products have become more mainstream and been adopted by, let's say, longer established companies, then the standards for quality, reproducibility, and efficacy have had to improve over that time.
I think that's a very good point, actually, that you make in terms of the, let's not call them new entrants to the markets, but the larger chemical companies have seen the opportunity in microbials and in biologicals in general, and they've started to work in that area. And of course, they bring with them a whole formulation history, so they understand what can improve the efficacy. And I think that is certainly helping.
Yeah, and so I think the early adopters are always going to try new things out. But of course, for the market to increase and develop, then we don't just need early adopters. We kind of need mainstream people adopting these products who then would be accustomed to, industry standards. And we have to make sure that they're getting products to those standards as the market develops. One of the things that we do spend a lot of time doing, and we have spent a fair bit of time doing when we've been drawing up reports or doing assignments for clients, for instance, is looking at what innovations have happened. So we might look through patents or publications. So what do you think has been impressive about innovation, or what need is there for future innovation in formulation in this area, do you think?
Well, I think, as you'll point out, when we look at a number of our challenges that we do work on, the word stability comes out a great deal. And one of the approaches to stability or improving the stability of microbials has been to use encapsulation. And I think the industry is catching on and has a great opportunity with some of the drives from other industries. So the big question being posed on encapsulation at the moment is the one of microplastics, and how are you going to actually encapsulate without generating microplastics? And of course, the way to do that is to use the natural and biodegradable ingredients, and that fits really well with what microbials and organic farming, etc., is trying to do. So I think that all of those innovations from other industries can be applicable to microbials, and I think to keep an eye on that would be beneficial for everybody who's active in this area.
Yeah, it's been noticeable actually that as these sorts of microbials are being formulated for agricultural uses, other industries such as probiotics, pharmaceuticals, even cleaning products have seen living microbial based products being introduced into the market, and they face many of the same sorts of challenges in terms of viability and stability that you would expect to see in the agriculture area as well.
That's right, and that again comes back sounding like a broken record to it's no different to formulating with ingredients in other marketplaces, and that's where we've made a living for the last 12 to 13 years to a large degree, is understanding what other people are doing and how that can be applied to specific market segments.
Sure. So I think that's really set the scene quite nicely, that little chat, and thanks for that. For me to then to hand over to you to give some examples of how microbials can be formulated. So I'm going to stop sharing my slides and let you take over at this point.
Okay, I'm just going to get to that, and then I'm going to share my screen. So can you see that screen?
I can, Jim? Yeah, that's fine.
Excellent. Okay, so thanks, Jim, for that chat. We thought it would be worth doing something a little bit different rather than bombarding you with PowerPoint, but we have to end up with bombarding you with PowerPoint. So away we go. If we think back to one of those early points, it's, well, what do you understand about your active ingredient? And that often comes back to how it's produced. And I think this is something that's often forgotten about. So I thought it was worthwhile starting with, well, how are microbial products produced? Not the formulations, the microbes themselves. And there are two ways. They're both fermentation. There's been a solid state, which we're not going to touch on today, but the most common one is a liquid state. So this that you can see here is really, to me, a stirred reactor. You have inlets, so you have a nutrient inlet, you have acid base if that's needed, and you obviously put things in there. You have measuring facilities, so you measure and control the pH, the oxygen, the heat, etc. You stir it, you have an aerator with oxygen, and then at the end of, well, you control the heat also obviously with a water jacket, and then you have something coming out the bottom. So this all starts, you inoculate with the typical inoculant is a microbe, some nutrients, which could be glucose, peptones, or yeast. Then in the fermentation process itself, you use that inoculant, you use more nutrients, you add in salts to control the environment. You may need an antifoam because obviously you're agitating it here. And you agitate, you aerate, and you control the temperature to get your optimum yield. So it's important to understand what you get. So what is the output from that fermenter that the formulator is going to be given? Well, that output will consist of a microbe. It will have some spores, depending on the type. It will have, very importantly, some metabolites. It will have still some nutrients left, and you may want to leave them in, or you may want to take them out. Again, your colleagues who are doing the fermentation will be able to tell you what can and what can't be controlled in here. As with every process, you will have some impurities and you'll have some water. So what can you do with this? I'm going to call it this raw soup. Well, post fermenter, you can allow it to settle if you've got the time to do that, or you can centrifuge to try and get the solid particles coming out. You then may decide that you want to wash it to remove some of the impurities or maybe even some of the nutrients, and then you may want to concentrate in some form or another. That could end up being what you call the fermenter broth. And this could actually be direct from the fermenter, or it could have been doing some of those refining processes that we've talked about. So what have people been doing with this fermenter broth in terms of approaches? Well, if they've decided that they're looking for a powder, they may mix the output with a carrier. And you're saying, well, what's the carrier? Well, fairly traditional stuff such as talc or kaolin has been used. They then will dry, and we'll come to drying over the next couple of slides. They then will sieve to deliver a fine powder which flows well and doesn't give any issues. And then they've added some adjuvants, and as we've spoken about it, the co-formulants and adjuvants, there's a good crossover there. In the early days, those adjuvants have been fairly simple and fairly limited in their scope. People have looked to make granules and have made granules, and again, it's mixing the output with a carrier, such as corncob grits, or in some cases, some clays. You then add the binder to bind the spores or the metabolites to the actual carrier, and then you'll dry and granulate. Again, we're going to touch on drying in the next couple of slides. If you've wanted a liquid product, then there's the general approach has been to use a suspension concentrate, SC, as you may know that. And as you've got solid particles, those spores, those microbes, you need to keep them dispersed. So you'll add in dispersants, and they will be from the same type of families that you have for dispersants for traditional SC formulations, but obviously they must be compatible. If you're making an SC, you all will probably know, well, I'm going to have to add in a rheology modifier, again, to keep those particles buoyant and to avoid them from settling. If it's water, you may have to add a preservative. Obviously, you need to be careful that it's not something that's going to kill the microbe. What you mustn't do, and the big difference when you've got a suspension concentrate of a microbial compared to a suspension concentrate of a typical chemical, is you mustn't dry mill it. You mustn't wet mill it. You mustn't mill it. Don't show it a mill. It's obviously, in a normal one, you're milling down to a particle size, but in terms of the microbe, as soon as you subject it to the shear and the forces in a mill, you're going to destroy it. So do not mill a microbial SC formulation. So I said we'd touch a little bit on drying. So let's talk about how people have dried their output. And because of the belief that temperature is a major issue to the viability of microbials, freeze drying has been used a lot. And in this process, moisture is removed through sublimation. So the material tends to be frozen and the moisture is removed under vacuum. So you're not necessarily subjecting it to any heat. Big advantage of freeze drying, it's very high yield. What you put in, you tend to get out, and you tend to get out almost all of what you did. It's very sterile, and it's used therefore a lot in pharmaceuticals, and it preserves the structure, which, if you're thinking about crystalline proteins, the structure determines how well they function to a large degree. So that preserving of the structure is extremely useful. However, freeze drying can result in some very long drying times. And if you're interested in output and economics, then that's not necessarily a good thing. And not only that drying time, but also the kit can be expensive when you think about the volume that you can get through freeze drying. And that is also because it's difficult to scale up. Every process, to be fair, is difficult to scale up, but freeze drying to scale up to the volume that if this market wants to achieve great penetration it needs is going to be quite difficult. But what do you mean by freeze drying? And I was at a training course last week where Sonny Clint Anderson from Janssen Pharmaceuticals put up this slide, and I thought it's worth sharing in a way. In the pharmaceutical industry, they freeze dry in vials. But you can see that they do the freezing and annealing, and then you get drying going on, and then at the end you get some secondary drying. But it's worth looking here at time in hours. To get to the secondary drying, you get 55 hours. That's a long time to get your product dry, and you only have a small volume. This is vials. They do lots of vials in a rack, but you can do bulk freeze drying. But only limited, obviously, because of the size of the film that you need to remove. Yeah, it just takes a long time. So what are the options? And for a long time, I think people have thought, I can't use hot air to dry my microbe. Well, that isn't quite true. And I was at this training course, ran this training course on spray drying last week, and there's a lot of expertise in this area. And the bulk of this, the main part is the spray chamber, but before that you have a product feed vessel, you have an air feed vessel. This is a co-current spray dryer, so both the hot air and the feed are sprayed in, and the spraying is important. You can use a nozzle, or you can use an atomizer, spinning disc atomizer or rotary atomizer, to actually generate the spray droplets that then get dried in the spray chamber. You can control the heat of that gas. You can control, and you don't necessarily have to heat up your feed. As it tumbles down the chamber, it is getting dried. It then comes out the bottom, it's collected in a cyclone, and you remove the fines as you would with any sort of powder product. So in a co-current, they both enter at the top. The highest driving force, and I'll show you a very interesting graph, is for drying at the top. These things dry extremely quickly in the top of the column. The powder and the air exit from the base. It's low viscosity. You can get very, very spherical particles from a spray dryer. You can get a good particle size, 100 to 200 microns. There is an option to use a fluidized bed at the bottom of a spray chamber or as a separate process, but that's not necessarily what you might need for a microbial. And this process is used and has been used for a long time for temperature sensitive solutions, where you get rapid drying and there are low times at hot and wet conditions. So the fear that your microbe is going to be subjected to, hot gas at 100 degrees C, it sees that for a very limited time, if at all. So those low times are very important, and therefore you can maintain the viability of a microbe through spray drying, and also good for high water content products. So again, you can do that with diluting down your feed. This is a generic graph, but you have temperature on your x-axis, and you have almost distance traveled through the spray drying column on your x-axis, and temperature on your y-axis. And it shows really you can have hot air. The difference between your hot air and your slurry entering is greatest at the top. They then converge until they're roughly the same at the end. But obviously, what can you do? You can control this temperature of the hot air. You can control the temperature of your slurry droplets, and if you need it a little bit longer, you just need, in a way, to build a bigger column, which is easier said than done. But if you do the design properly, you'll be able to understand how you can minimize the temperature that your product will see. And I think that's very important. There is obviously capital cost in terms of spray dryers. There is a capital cost there, but you can increase your throughput significantly if you use a spray dryer. So as we're moving on, I thought we can't do a formulation of microbials without talking about Bacillus thuringiensis, or BT as it's known. We're going to talk about what is it, where it's used, what strains are being used commercially, what formulations are in there commercially. It's not a new microbe. 1902, first discovered in silkworms in Japan. It is the most commonly used microbe, bioinsecticide in the biocontrol market. It's a gram-positive microbe, which Jim spoke about at the beginning, and it produces these crystal proteins, often referred to as CRY toxins, during sporulation, and they do the heavy lifting. They are toxic to a wide number of larvae through ingestion, so they ingest in the toxins and then it destroys the larvae through from the inside. But they are different. There are lots of different ones, and you'd have to ask your microbiologist and your biochemist about this, but a number of different CRY proteins, and each of them are produced in different ratios. So again, trying to understand what you've got and the sensitivities, try and understand what you're getting from your fermenter. What is the ratio of those proteins? That gives it the chance to have multiple modes of actions. It also provides some differentiation between commercial products. So what are the subspecies? And this data has all come from regulatory databases in the US and Europe. You've got Izawi, you've got Israeliensis, and you've got the big one that we're going to talk about, which is Kerstaki, Tenebrionsis and Valensis. Kerstaki in itself, again, gets complicated, but there are a number of different strains, and they all are identified by their genes. So you can look at a product and it will tell you what strain is actually in that product, and that strain will differentiate it from other commercial products. How has BTK been formulated? It's been formulated, well, we spoke about it earlier. It's been formulated as a dry powder, as a wettable powder, as a water dispersible granule, AWDG, also as a dry flowable. We see products that are marketed as a suspension concentrate, and then some products marketed as an emulsifiable suspension, which is in essence an oil dispersion. So it's a suspension concentrate, not using water. So this area of oil dispersions is also becoming quite attractive to the microbial market, because where you do have an issue with water sensitivity and water activity, if you use oil as your media, then you remove that a great deal. The issues mentioned when you formulate these products, well, you've got shelf life, you've got spreading, sticking, you've got UV resistance, you've got rain resistance, but we can go and look at the traditional crop protection market, and these issues are also mentioned, and a lot of them are solved by the use of co-formulants and adjuvants. Just flicking through a few of the selected commercial products, Jim spoke about Dipel from Valent. That's the subspecies, the strain, sorry, ABTS 351. Won't go through these. You have to dig a little bit to try and get some of the ratios, but that's roughly, you've got 4 different crystalline proteins, plus you've got a secondary product called VIP3A and svitamycin A. It's been formulated different brands as a WP, a WDG. It's listed on OMRI, the Organic Materials Research Institute, and there's also been ES with the Dipel brand. If we move to another brand from Valent, you can see it uses a different strain, and there are some different crystalline proteins and different ratios. So again, you can see how that will indicate where it will be effective and may indicate what the formulator needs to understand. Dry flowable, water dispersible granule, and OMRI listed. Foray from Valent, another grade, is an SC, which is again is OMRI listed, aimed at forestry and ultra low volume. And I think that's an interesting area. The ultra low volume and delivery by drones is another technology that's being looked at more and more and being used in agrochemicals. And that may be an opportunity, or maybe an area that microbes need to look at. The SC, they say, contains carriers, suspension agents, and stabilizers. Well, we wouldn't be surprised about that because it is an SC formulation. Just so you don't think Valent are the only ones around with BT, Belthirol from Pro Belte, again, a different one. It's a WP, and when you look in the literature, it says it's protected against degradation by UV light. So maybe they've done a little bit of encapsulation. Maybe they put an additive into their powder to actually protect it from UV light. They also have an SC and a dustable powder. So those are the commercial products that are around. I thought, just to give you a little insight into some of the things we've seen by looking at patents with BT formulation to address that UV resistance. There is a patent, you can look it up yourself, 2024, assigned to the University of New Mexico, Rainforest Innovations. And what they've done and many people, as I've said, have approached this through encapsulation. They've looked at encapsulating BT with cross-linked chitosan or alginates. And that's a technology that's well known in other areas, but hasn't necessarily been applied and given the surprising benefit in the patent of UV resistance. So that's an interesting one to look at. Also looking at UV, and in this case, rain resistance, there was a patent in 2021 assigned to Dow Global Technologies and Roman Haas at the time, but Dow Global Technologies. And they claimed that you could add in glycols, which are commonly put in as humectants, and polyphenols into the same formulation. And they claimed that normally when you put those two together, they're incompatible. But they said that when you put it in with BT, solid or liquid, and using a specific molecular weight of glycols, then you didn't get that incompatibility, and you also got improved UV and improved rain resistance. So there's a lot more around, and I'm sure many of you in the audience have looked around the patent literature and the academic literature that we just simply don't have time to do today. I don't know, Jim, if you want to join, but I haven't seen any questions. We're close to time as it is.
I've got one. There's one question here from Shazia, which is a really good question, I think. Which is a really good question. I think you're welcome to anybody else to add any further questions you may have. But Shazia asks, we stop unwanted microbial growth while also keeping our biologicals alive in our formulation? This is a really key question. And secondary question, how much preservatives is too much or enough? I guess to start with, David probably got some further answers, but I would say don't have water in the formulation would be an easy way of doing it, and formulating a dry product like a granule or a powder, or using an oil-based suspension rather than a water-based suspension, and that could be an oil that is immiscible in water and emulsifiable in the tank, or it could be using a fluid such as a glycol, which is water miscible as a suspending fluid. So a number of different ways of doing it. David, have you got any other thoughts on?
Yeah, I mean, water is an issue, but there is a thing called water activity. So sometimes if you can lower your water content below what's called the critical water activity for your microbe, then that will help a great deal. In terms of how much preservative is needed, Jim, you're the preservative expert.
Well, I think one of the challenges is that a lot of the preservatives which are used for formulating sort of water-based suspensions are quite broad spectrum in their activity. They will control almost all fungi and bacteria. So finding a preservative that is not actually going to kill your active is quite challenging. So you can do challenge testing, and you can choose a concentration. If you need a preservative, that is, you can choose a concentration that stops growth but doesn't kill the organisms off. So you can do those sorts of tests, a microbiological test, to choose the preservative level. Or you could move towards a system, if you've got water in it, that is, that contains, it doesn't contain a preservative at all, but contains a component that will reduce the water activity, as David said. So it might be a humectant type molecule, a glycol or something that would bind the water and reduce the concentration of free water to keep it below that, the water activity below the critical level. So I think the answer to the second question is you have to test for it and do the right microbiology testing at a range of different concentrations.
Yeah, I think that's fair comment, and yeah, unfortunately, sometimes you just need to test. There isn't that magic answer that we can give in terms of
a magic number. No. Welcome any final chances for questions, David? Do you want to just round off?
Yeah, I mean, you know, you're welcome to contact us via email on info@iformulate.biz, or david@iformulate.biz, or jim@iformulate.biz. I know, looking at the attendee lists, we know many of you in the audience, so please just come and ask. This is you've been led into a prompt for what we are developing now, which is a new online course on the formulation of microbials. Our present plan, we occasionally have a plan, is that will be ready for delivery in September or October. If you are interested in that, then email us and we'll send you more information when it becomes available. You will be able to see it on our training and events page, which is on that link, and you're obviously going to get those slides. You'll also be added to our newsletter list, which we only send out probably one a month, if that. And you can easily unsubscribe if you're tired of hearing from me and Jim and our anecdotes on what not to do and what to do. But with that, I think we're going to say thank you again for your attention. And yeah, we look forward to interacting further with you, Jim.
Yeah, thanks very much for your attention today.
Thank you. Bye-bye.